All right, so thank you very much for the kind invitation and introduction. It's our pleasure to be here. I'm David Jimenez Morales. I'm bioinformatics lead here at the MoTrPAC Bioinformatics Center. With me is Jimmy Sen, which also will talk, is the infrastructure lead at the Bioinformatics Center as well. We are going to talk today about MoTrPAC. We are going to talk about this comprehensive multi-studies, multi-omic, multi-tissue insights into exercise molecular responses. You are going to hear the word multi many times. So, but the first take-home message that we want you to take is that we are the consortium that studies exercise. Right? MoTrPAC, I want first to formally tell you that MoTrPAC stands for Molecular Transducer of Physical Activity Consortium, that our goal is to study the molecular changes that occur during and after exercise, and we aim to generate this molecular map of the effects of exercise and training. I guess that the question, you might be wondering, why are we studying exercise? And here you can see a list of all the benefits that comes with exercise. It's the possibly one of the best medicines that you can take. We would like to understand how physical activity protects against some of these chronic diseases that are listed on the screen, but also how it promotes overall health. So this is a multi-center study, many institutions all over the country that are part of this consortium. And that's why first we need to start by acknowledging all these people, and thanks to all the members of MoTrPAC, we are able to generate what we are going to show today. This is also a multi-omics study, and we are covering, I think, probably all the omics that are available. And this is also a multi-species study, yes, where we have humans and our neighbors from the last 10,000 years, right? So that they are also going to help with this.
Best friends.
Yeah, we're best friends. So we are also a multi-study consortium, everything about exercise. And with respect to, let me give you an overview. With respect to humans, we have multiple studies as well. The first one is that I'm going to mention is the acute exercise study, which is done on a relatively healthy cohort of human individuals, which are randomized to either be part of the endurance or the strength study or the control, and they are doing a single bout of exercise, and we do a number of biospecimen collection. Then we have another cohort of highly active individuals as well that are randomized to either do an endurance or a strength exercise. We will hear more about these details later. Another study is on these sedentary adults, which in addition to this bout of acute exercise, there are also a training component for 12 weeks. And also in endurance and training, right? So, endurance and strength training. So the biospecimens that we are collecting are blood, muscle, and adipose tissue for the adults. And we also have a study on pediatric, but this is only focused on endurance, both an acute study, but also a training component. And the biospecimen that we are collecting for kids is only blood. And then we have the rat studies, and here's a summary. Later we will talk more about these studies on rats. Basically, we are going to study two groups, two different ages. One are the 6 months, which are the equivalent to young adults, and then we have the middle-aged adults. And we are studying like acute exercise as well, like in humans, and also training and endurance training. And one of the key elements of studying rats is the biospecimen collection. We perform a number of analyses and trying to characterize the molecular profiles in many of the tissues, so we can study this response to exercise across all these tissues. That's the advantage of working with this animal model. Again, this is the overview. I encourage you to read this paper that we reference here, the Cell paper, where is the markup paper where we explain all the details about this project. So I encourage you to read that paper. So MoTrPAC is aiming to address key gaps in the field. Here's a summary of the main studies conducted, the Multi-Omics study conducted on exercise. And MoTrPAC is aiming to bring a lot in order to advance our knowledge on exercise. But let's begin with the first study that we have released, that is on endurance training. And there is nothing better than showing you the timeline and the study design, so you get an idea of what we did here. So we have obviously rats that were that exercise, and we are talking about 48 rats that exercise for not for 8 weeks. I will explain you how this works. We have the controls, and as you notice here, the timeline is in weeks, and each of these green lines represent the bouts of exercise that they did every day. After 1 week of training, 12 of these rats were sacrificed and all the biospecimens collected. And the rest of the rats remain running and exercising. After 2 weeks, we have another sacrifice point, and so far and so on. So as you might notice, the length of these bars are increasing, and this is because this is a chronic progressive training where we increased the inclination and duration and the speed of the treadmill. And also important to notice here is that there is a 48-hour rest or washout period. So that's why we have these gaps between the exercise bouts and the collection, the sacrifice time point. So because we want to make sure that we are only studying the effects of training, right? Something important to point out here, another detail that I want to provide is that this is how the data is collected. So, especially, the goal here is that you to understand the amount of work that is behind this study. After 1 week of training, these are the number of rats that are sacrificed at each time point. And for each rat, we are collecting all these different tissues. And for each of these tissues, we are doing all these sums. We are covering like transcriptomics, epigenomics, by 2 different chemical analysis sites, 2 different labs, one in Stanford, another one in Mount Sinai. We have Proteomics doing protein abundance, phosphorylation, acetylation done at the PNNL and the Broad Institute, and then we have a large panel of metabolomics assays. We have untargeted metabolomics done at the Broad Institute, Michigan, or Georgia Tech, and then we have the targeted assays done at Duke, Mayo Clinic, and Emory. So, but we didn't do all the assays to all the tissues, but here we are summarizing what it was done in each of the tissues for this particular study, and we, as you can see, heart and liver got all of them. And then we have other tissues that got a lot of other different assays. We're talking about white adipose or skeletal muscle, kidney, lung. All that got a important amount of assays where we were trying to characterize the molecular changes. So and now I'm showing here what you can see on the screen is a summary of the number of training regulated features. Let me explain this. On the left, you see each of the rows represent assay from each of the OMs. We have the RRBS for the methylation, ATAC-seq to characterize the chromatin accessibility. We have transcriptomics, we have proteomics, phosphorylation, so far and so on, right? And importantly, the background color of the numbers is representing the differential proportion of analytes. So as you can see, for example, if you take a look at the epigenomics part, we didn't detect that many changes. relative to the number of peaks that we detected. But when you look at the transcriptomics component, you see that there is a huge variety of responses depending on the tissue. For example, look at the adrenal gland, and we have it was the largest number of differential transcripts that we were able to detect, and then followed by colon. And then you see the adipose tissues, white adipose, brown adipose. You see large number of differential transcripts, and then blood. And then we have other tissues that we barely detect any change, like for example, in some of the brain tissues, like in cortex or hippocampus. And you know, this variety of responses is also captured in the protein abundance assay. But some of these results, I mean, everything was, this study has been published. It was published like a few months ago, as you can see in the reference in Nature. It was featured in the cover. It's something that we are obviously very proud of. What we can do is to give you, you know, it's a huge paper, but we can give you a few of the biological findings and a few of the highlights. For example, with respect to the phenotypic changes after 8 weeks, of course, we detected an increased aerobic capacity, the VO2 max, approximately almost at 20% in both males and females. Interestingly, the body fat decreased only in males, while increasing in the controls. And the body weight increased in females in all time points, and no changes, no significant changes were detected in males. Then we had like thousands of shared and tissue and sex-specific molecular changes. And some tissue remodeling patterns were observed, especially in the immune system, in metabolism, and in heat shock stress response, also in the mitochondria or mitochondrial pathways. And these changes that we observed were consistent with some of the changes that has been characterized in humans. Something very interesting were the sex differences of the effects of training, especially in adrenal gland, in lung, or in adipose tissue. Very interestingly as well, we found potential links between some of these training adaptations and some other diseases, like for example, the non-alcoholic fatty liver disease, or inflammatory bowel disease, or cardiovascular health, or tissue injury recovery related pathways. So, you know, the Nature paper was like the landscape paper, but there are a number of companions, and we want to give you a few also biological highlights about some other papers. For example, this paper that was published in Cell Metabolism. And a couple of things to comment about this paper. One is tissue-specific mitochondrial adaptations that we were able to characterize. Endurance training induced substantial changes in mitochondrial analytes across multiple tissues in rats. The response to training was it varied depending on the tissue, highlighting the tissue-specific nature of the mitochondrial adaptation. In muscle, we were able to characterize an increase in oxidative proteins, which are important for preventing age-related decline. The level, and in liver, we were able to characterize a shift in lipid metabolism, which is potentially driven by the increase in acetylation of metabolomic proteins. With respect to the disease relevance, it was very interesting to notice that to find that some protein networks that were, for example, upregulated in exercise were downregulated in some other diseases, like for example in diabetes or cirrhosis. For example, it was very interesting in the case of this particular protein, HSD17B10.
I remember that.
Yeah. Which was identified as a central regulator in both diabetes and cirrhosis. For example, it was upregulated with exercise and downregulated in disease. Very interestingly, the knockdown of this protein elevated the oxygen consumption in a human liver cell line. And as you might imagine, this has implications for mitochondrial dysfunction and metabolomic stress.
David,
But these are not
I pause you for 1 second?
Yeah, sure.
There's a question in the chat from a couple slides ago, I think. It's from Doug Keel. It seems peculiar that the skeleton is excluded from all the molecular assays. Is there some rationale for this, given bone's exquisite sensitivity to exercise? That's a good question. What are your thoughts?
The skeletal muscle, we are including skeletal muscle. Which one? Vastus lateralis is the one that we decided to start?
Skeleton bone. Yeah. So there's concerns about being able to process the bone samples at the throughput necessary for the study. But there are bone samples stored in a biorepository for future analysis.
Yeah.
Oh, very good.
Yeah. That was Matt Wheeler, just in case that you guys couldn't know.
Yeah, Matt moving, still answering questions. I love it. A question from Henning. Were the rats free to feed as much as they wanted, or were they restricted to calories?
I think that they were not restricted, as far as I remember. I mean, they were controlling the amount of food that was provided, but I don't remember from the top of my head if what was.
Yeah, there was no calorie restriction. There was a period of removing the food before sacrifice that was standardized, but there's no calorie restriction during this training.
Okay, we can keep going. Let's go back to where we were. Yes, we were mentioning that you have all the publications available. The ones, all of them are open access. So please go to this page and download all the publications. And thanks to this study, we are building a number of resources which are available to the scientific community. Obviously, an important one is the data. You can download all the data through the MoTrPAC Data Hub. We will talk more about how to get access to the data in a moment. Jimmy will walk us through the MoTrPAC Data Hub, which are mostly making available the results, different type of results. But then we have all the data also available in public repositories. If you are interested in analyzing the raw data, you can go to these public repositories. We are also making available all the source code and software that we have developed. For example, so we are trying to make everything transparent, from data injection, all the code to for the pipelines that we develop for the identification and quantification, or everything was developed in close collaboration to each of the labs that are part of the consortium. We have source code for quality control, extensive data analysis, and also code available. Very importantly, we made available some R packages, and this is a very nice feature, and also something that should be pretty useful. So in most of our users in the consortium use R, so that's why we decided to put everything in R packages. And it's as easy as install that data library that we are listing here, and you will have all the differential abundance results and all the normalized tables for you to do your own analysis. But if you need some extra functions, we also built another library that helps you do some analysis on our data sets. You will be able to reproduce the results that we have in the papers, right? For example, I don't know, if you want to do a PCA, we'll help you with that in that library. They are nicely documented, so we encourage everybody to check it out. So there are more. So for each of the publications, every time. So we are trying to make our packages to facilitate the exploration of the data. Again, we want the world to use our data and trying to help him unveil and anything that could help us understand how exercise benefits us, right?